Welcome to the deep dive. Today we're getting into something really fundamental. How our genes actually get used,
Yeah, we're talking about the control of gene expression.
which is basically how the instructions packed away in our DNA are actually, read and used to build things.
Yeah.
Mostly proteins, right? With RNA as the go-between.
Exactly. And you might think, why should I care? But honestly, it's super relevant. Just think about all the different cells in your body.
Oh yeah, like neurons buzzing away, immune cells fighting stuff off.
Lymphocytes, yeah, or pancreas cells making hormones, red blood cells carrying oxygen. They look totally different, do completely different jobs.
It really is mind-boggling. A brain cell versus, say, a liver cell, worlds apart.
And here's the kicker, the real puzzle.
They all have the exact same DNA blueprint inside.
Right.
Almost every single cell. How is that even possible?
That's the core question. The answer is this amazing, intricate, system-controlling gene expression.
So it's about which instructions get read in which cell and when.
Precisely. Which genes are switched on, making their RNA and proteins, and which ones are kept quiet. That's what we're diving into today. We'll unpack the mechanisms, kind of give you a shortcut to understanding this biological cornerstone.
And our sources have some pretty detailed stuff, even visuals.
Yeah, they really lay out the evidence and the processes involved. It's fascinating stuff.
Okay, so first things first, this idea that all cells have the same DNA. How do we actually know that? I mean, surely a muscle cell doesn't need the instructions for, like, making eye pigments. Did it just toss them out?
That's a really logical thought. But some clever experiments, especially cloning, proved otherwise. Remember that frog experiment?
Vaguely. Involving a skin cell.
Yeah. Scientists took the nucleus, you know the bit with all the DNA from an adult frog's skin cell, a specialized cell.
Okay.
Then they popped it into a frog egg where they'd already removed the original nucleus.
Right, a nucleated egg. And what happened?
That egg, with just the skin cell nucleus, it developed into a tadpole.
Wow. Seriously.
Yeah.
A whole tadpole from just the DNA of a skin cell.
Exactly. The huge takeaway was that the skin cell nucleus must have still contained all the genetic instructions needed to make every type of cell in that tadpole. Muscle, nerve, gut, everything.
So it hadn't lost any genes along the way. It just wasn't using all of them in the skin cell.
Precisely. Similar things were shown with plants cloning a whole carrot from one root cell, and even mammals, like cloning a cow from an epithelial cell nucleus.
So the evidence points strongly towards specialization, or differentiation as scientists call it, being about changing which genes are expressed, not about losing the DNA itself.
You got it. Cell differentiation is about differential gene expression.
Okay, so if they have the same DNA but do different things, they must be making different stuff, like different RNAs and proteins. Can we actually see that difference?
We absolutely can. There are techniques designed specifically to visualize this.
Okay, now this sounds cool. How do we peek inside and see which genes are, like, switched on?
One really powerful tool is the DNA microarray. Imagine it like a heat map for gene activity.
A heat map.
Yeah, so each row might represent a different gene, thousands of them, and each column is a different sample, maybe different tissues like liver, brain, muscle, or even different types of cancer cells.
And the colors tell you.
What? The color in each little square tells you how much messenger RNA or mRNA is present for that specific gene in that specific sample. mRNA is that temporary copy of the gene that's used to build the protein.
Oh, okay.
So typically, red might mean lots of mRNA, the gene is highly active. Green might mean very little, the gene is quiet. And black, somewhere in between, like average levels.
So you look across the grid and see, like, this gene is blazing red in liver but green in brain.
Exactly. And you see these really distinct patterns. Liver samples have one signature of red and green across the genes. Brain samples have another, muscle another. It's a visual confirmation that different tissues have unique profiles of gene activity.
Wow. So distinct patterns of gene expression for each cell type. Got
Yeah.
it. Is there a way to look at the proteins directly?
Yep. Another technique does just that. It's called 2D gel electrophoresis. It separates out all the proteins in a cell extract based on two properties.
Two properties. Like what?
First, their electrical charge, and second, their size or mass. So you end up spreading the proteins out across a gel.
Creating a kind of protein fingerprint for the cell.
That's a great analogy.
Uh-huh.
Our source shows a comparison. I think it was brain versus liver cells. You see spots all over the gel, each spot being a different protein.
Okay.
Some spots are in the same position on both gels, maybe colored red in the example. Those are proteins found in both brain and liver. Probably essential housekeeping proteins needed by most cells.
Right, the basics.
But then you see other spots, maybe colored blue, that are only on the brain gel or only on the liver gel. Those are the unique specialized proteins that give each cell type its specific function.
Ah, so we can literally see the different protein toolkits the different cells are using.
Exactly. So the evidence is solid. Same DNA, different RNA and protein products because of differential gene expression. Which brings us to the big question. How does the cell control all this?
Okay, let's get into the control mechanisms. Where does this regulation actually happen? On the journey from DNA to a working protein, where are the switches?
Well, it's a multi-step journey, and the cell actually has control points at almost every step. Our source lays them out nicer.
Yeah, what's the first big one?
Transcriptional control. This is deciding if a gene should be transcribed into RNA in the first place, and if so, how often. This is often seen as the main control point.
Why the main one? Just efficiency.
Pretty much, yeah. If you don't even bother making the initial RNA copy, you save a ton of energy and resources compared to making it and then deciding not to use it later.
Makes sense. Don't start the process if you don't need the final product.
Okay.
So transcription is number one. What else?
After the RNA is made, there's RNA processing control. That's things like how the RNA is spliced, capped, and has a tail added, all happening inside the nucleus.
Splicing. Right, where they cut out bits. Okay.
Then there's RNA transport and localization control. Which completed RNAs get shipped out of the nucleus, and where do they go in the cytoplasm?
Ah, directing the messages.
Next up is translational control. Once the mRNA is out in the cytoplasm, should it actually be translated into protein by the ribosomes, and how quickly?
So you can have the message sitting there, but not actually make the protein yet. Interesting.
Definitely. Then there's mRNA degradation control. How long does that mRNA message stick around before it gets broken down? The lifetime varies a lot.
Right. A short-lived message means less protein over time.
And finally, even after the protein is made, there's protein activity control. The protein might need to be switched on or off, modified, or even deliberately degraded when it's no longer needed.
Wow. Okay. That's a lot of control points. Transcriptional, processing, transport, translational, degradation, protein activity.
It really is a multi-layered system. But like we said, that first step, transcriptional control, is often the most critical gatekeeper.
So let's focus there for a bit. How does the cell make that initial decision? Transcribe this gene or leave it alone?
It largely comes down to proteins called gene regulatory proteins, or sometimes transcription factors. These are the key players. They recognize and bind to a specific short sequences of DNA called regulatory DNA sequences, which are usually located near the gene they control.
So these proteins are like molecular switches that find the right spot on the DNA.
That's a good way to think about it, yeah.
Yeah.
They act as activators, turning genes on, or repressors, turning them off.
How do they find the right spot? DNA is huge.
It is. To understand that, you have to picture the DNA double helix structure. It has grooves, right? A wider major groove and a narrower minor groove. Most gene regulatory proteins make contact with the edges of the base pairs in the major groove.
Why the major groove specifically?
It's wider and exposes more features of the base pairs. This allows the protein to make more specific chemical contacts, to essentially read the sequence without having to unwind the DNA helix. Often, a part of the protein, like an alpha helix structure, fits right into that groove.
Okay, it fits in the groove. But how does it recognize, say, a specific sequence like GATTACA?
It's all down to chemistry. Specific amino acids on the surface of the protein form bonds with the specific base pairs exposed in the groove.
What kind of bonds? Like strong connections?
Not usually individually strong, no. They're things like hydrogen bonds, ionic bonds, and hydrophobic interactions. Each single contact is quite weak.
Weak. How does that work then? Wouldn't it just fall off?
But the key is that a typical regulatory protein makes many of these contacts simultaneously, maybe 10 to 20 specific interactions with its target DNA sequence.
Ooh, okay. So lots of weak bonds add up to a strong and, more importantly, specific interaction, like a very precise molecular handshake.
Exactly. That collective binding energy ensures it sticks tightly only to its correct target site and not to the millions of other random sequences in the genome. The specificity is incredible.
And these target sequences themselves, do they have any common features?
Often, yeah. Many recognized sequences are palindromic, meaning they read similarly forwards and backwards on opposite strands, or they have repeated elements. This often relates to how the regulatory proteins themselves bind, frequently as pairs or dimers.
Right, so these proteins need the right 3D shape to fit the DNA and make those contacts. Are there common structural designs they use?
Yes, absolutely. Evolution has converged on a few successful structural motifs, kind of like modular keys that proteins use to bind DNA.
And you mentioned they often work as pairs, dimers. Why is that?
Dimerization, yeah, it's very common. Working as a pair can significantly increase the binding strength and also the specificity. Plus, if you can mix and match different subunits, heterodimers, you can recognize an even wider range of sequences.
Clever. Okay, so what are some of these common DNA binding motifs?
One of the classics, found very early on, is the helix-turn-helix, or HTH motif.
Helix turned helix. Sounds descriptive.
It is. It's basically two short alpha helices joined by a short loop or turn of the polypeptide chain. One of those helices, usually called the recognition helix, fits snugly into the major groove and makes those critical sequence-specific contacts.
And the other helix helps position it.
Sort of, yeah, helps stabilize the structure. This motif is really widespread, found in bacteria, eukaryotes, everywhere. The TREP repressor and LAC repressor we might talk about later use it.
Okay, HTH. Got it. What's another one?
A related but distinct one, especially important in development, is the homeodomain motif.
Homeodomain. I've heard that in relation to Hox genes.
Exactly. Homeodomains are characteristic of the proteins encoded by homeotic selector genes, like Hox genes, that control body plan development. Structurally, it's made of three alpha helices packed together. Helix number 3 usually acts as the recognition helix in the major groove.
So, similar principle to HTH, but a bit more complex with the three helices. Okay. What else? Zinc fingers.
Ah, yes. Zinc fingers are very common, especially in eukaryotes. As the name suggests, they incorporate one or more zinc ions as structural components.
The zinc holds it together.
Precisely. A common type uses a zinc atom to coordinate an alpha helix and a beta sheet structure. The alpha helix part often lies in the major groove, reading the DNA sequence. A cool thing is that many proteins have multiple zinc fingers linked together, allowing them to bind longer, more specific DNA sequences.
Like reading a longer word on the DNA.
Exactly. And they often dimerize too.
Okay, zinc fingers rely on zinc. What about leucine zippers? Sounds like clothing.
Huh. Yeah, the leucine zipper motif is formed by two alpha helices, typically one from each protein in a dimer. These helices have hydrophobic amino acids, often leucine, at regular intervals.
Leucines.
These leucines interlock, kind of zipping the two helices together into a coiled coil structure. Extensions from these zipped helices then spread out, forming a Y shape that grips the DNA, usually in the major groove on opposite sides.
A literal zipper holding the protein dimer together while it grabs the DNA. Cool. One more. Helix loop helix.
Right, the helix loop helix, or HLH motif. This one features a short alpha helix connected by a flexible loop to a longer alpha helix.
Short helix, loop, long helix.
Yep. Like leucine zippers, they usually function as dimers. The longer helices are often involved in the dimerization, interacting with the long helices of the partner protein, while the shorter helices tend to make the specific contacts with the DNA. These are also really important in controlling cell differentiation in eukaryotes.
Okay, so a whole toolkit of structural motifs, HTH, homeodomain, zinc finger, leucine zipper, HLH that proteins use to latch onto specific DNA sequences. But how do scientists actually figure out which protein binds to which sequence and where on the genome?
Excellent question. We need ways to study these interactions. Our sources mention several key techniques. One is the gel mobility shift assay, often called EMSA.
Gel mobility shift. What shifts?
The idea is simple. A fragment of DNA will move through a gel matrix at a certain speed when you apply an electric current. But if a protein binds to that DNA fragment, the complex is now bigger and bulkier.
Ah, so it moves slower through the gel. Gets shifted.
Exactly. You run labeled DNA fragments on a gel, some mixed with protein extract, some without. If you see a band that's higher up, moved slower, in the lane with the protein, it indicates that a protein bound to the DNA. It's a basic test for binding.
Molecular traffic jam on the gel. Nice. What else?
Another approach is DNA affinity chromatography. This is more about purifying the protein that binds to a specific DNA sequence.
How does that work?
You attach many copies of your DNA sequence of interest to beads packed in a column. Then you flow a whole mixture of cellular proteins over the column.
Okay.
Most proteins will just wash straight through, but if a protein specifically recognizes and binds to your DNA sequence on the beads, it'll stick to the column.
Like fishing for the protein using its favorite DNA bait.
Precisely. Then you can wash away all the non-binding proteins and finally change the conditions, like increasing the salt concentration, to release your target protein from the DNA, hopefully in a much purer form.
Clever. What about finding the exact spot where the protein binds on the DNA?
For that, there's DNA footprinting. It's really elegant.
Footprinting.
Yeah, you take your DNA fragment, labeled at one end, you let your protein bind to it, then you lightly treat the mixture with an agent that cuts the DNA, like DNA's eye, but under conditions where it cuts randomly and, on average, only once per molecule.
Okay, so it snips the DNA strands.
Right, but here's the crucial part. The places where the protein is sitting, physically covering the DNA, are protected from being cut.
Ah, the protein shields its binding site.
Exactly. So when you remove the protein and separate the resulting DNA fragments by size on a gel, you'll see a ladder of bands representing cuts at almost every position, except in the region where the protein was bound. That region appears as a gap or a footprint in the ladder.
It literally leaves its footprint on the DNA sequence map. That tells you precisely where it sits. Very cool.
It is. Now, what if you have a protein and you don't even know what DNA sequence it likes to bind to?
Yeah, how do you figure that out?
There are methods like SELEX or simile-systematic evolution approaches. You start with a huge pool of short DNA molecules, each with a different random sequence in the middle.
A massive library of random DNA words.
Pretty much. You mix your purified protein with this pool. The protein will bind to some of these random sequences, presumably the ones that best resemble its true target site.
Okay.
You then isolate the protein DNA complexes, maybe using that affinity method, discard all the unbound DNA, and amplify the bound DNA sequences using PCR.
So you enrich for the sequences the protein actually liked.
Exactly.
Yeah.
And then you repeat the cycle. Bind the protein to this enriched pool, isolate the bound ones, amplify again. After several rounds, you end up with a population of DNA sequences highly enriched for the protein's preferred binding site. Then you just sequence them to figure out the consensus binding sequence.
Like directed evolution in a test tube, selecting for the best DNA binders.
Precisely. And one more really important one, Chick-pea, chromatin immunoprecipitation.
CHOCORP. What does that tell us?
Chick-pea is powerful because it tells you where a specific protein is bound to DNA inside a living cell in its natural context, including the chromatin packaging.
Whoa, in the cell. How?
You first treat living cells with chemicals like formaldehyde that cross-link proteins to nearby DNA. Basically, you freeze everything in place.
Okay. Gluing the proteins to the DNA they're touching.
You just break the cells open and shear the DNA into smaller fragments. Now comes the key step. You use an antibody that specifically recognizes your protein of interest.
Oh, antibodies to grab just your protein.
Right. The antibody pulls down your protein, and because it's cross-linked, it brings along the DNA fragments it was bound to. You then reverse the cross-links, purify the DNA, and identify those DNA sequences, often using sequencing, Chikusec.
So you get a map of all the locations in the entire genome where your specific protein was sitting in the original cells.
Exactly. It's incredibly informative for understanding gene regulation in its native environment.
Okay, so we have amazing tools to study protein-DNA interactions. Now let's think about how this regulation actually plays out. Our source says things are generally simpler in bacteria, prokaryotes.
That's definitely true. Bacterial gene regulation has some key differences. One big one is the use of operons.
Operons, right. What are those again?
An operon is basically a set of genes that are right next to each other on the chromosome, are involved in the same pathway, like making an amino acid or breaking down a sugar, and are all transcribed together into a single mRNA molecule from one promoter.
So it's like a package deal. One switch controls the whole set of tools needed for a specific job.
Exactly. It allows for really efficient, coordinated regulation. Turn on the operon, you get all the enzymes you need. Turn it off, you stop making all of them.
Smart. And the main players controlling these operons are?
Primarily repressor proteins, which bind to DNA, usually at a site called the operator near the promoter, and block transcription. They turn genes O off F. And activator proteins, which bind to DNA and help RNA polymerase start transcription, they turn genes on.
Okay, repressors off, activators on. Our source uses the tryptophan operon. the TREP operon, as a classic example of negative control. How's that work?
Right. The TREP operon has the genes for making tryptophan. The control system is elegant. There's a TREP repressor protein. When tryptophan levels in the cell are low,
The cell needs to make more tryptophan.
correct. And in that low tryptophan state, the repressor protein is inactive. It can't bind to the operator DNA sequence.
So the track is clear.
Exactly. RNA polymerase can bind to the promoter and transcribe the operon genes. The cell makes the enzymes and thus makes tryptophan.
Okay, simple enough. But what happens if the cell has plenty of tryptophan? Maybe it got some from its environment.
Ah, now the system kicks in. When tryptophan levels are high, tryptophan molecules themselves bind to the TRP repressor protein.
Tryptophan binds to its own repressor.
Yes, tryptophan acts as a copressor. Binding tryptophan causes the repressor protein to change its shape. This is called allostery.
It changes shape.
And now the repressor, in its tryptophan-bound shape, can bind tightly to the operator DNA sequence.
Ah, and that blocks the promoter.
It physically blocks RNA polymerase from binding or moving forward. So transcription of the TRP operon stops. The cell stops making the enzymes to synthesize tryptophan because it already has enough.
That's a beautiful feedback loop.
Yeah.
The end product, tryptophan, shuts down its own synthesis pathway when it's abundant. Very efficient.
It really is, and this illustrates the basic idea of negative regulation. The gene is on end by default unless a repressor turns it off.
Our source mentioned induction and corepression.
Right. What we just described with tryptophan is corepression. The repressor only binds DNA when the small molecule tryptophan is present. Induction is the opposite, like with the lac operon. There, the repressor binds DNA and keeps the gene OFF unless a small molecule, allolactose derived from lactose, binds to it, causing it to fall off the DNA, turning the gene on in.
Okay. And positive regulation is the flip side. The gene is OFF unless an activator turns it on.
Exactly. An activator protein might need to bind to the DNA to help RNA polymerase get started. Sometimes the activator itself needs a small molecule signal to become active and bind.
And some regulators can even do both, activate or repress.
Yeah, dual function regulators exist. The lambda repressor from a bacteriophage is a classic example. Depending on exactly where it binds relative to the promoter, it can either help or hinder RNA polymerase. Context matters.
Okay, let's talk about that other famous bacterial example. The lactose operon or lac operon. That one's a bit more complex, right? Has both negative and positive control.
It does. It's a fantastic example of integrating two different signals. So the lac operon has genes for breaking down lactose, the sugar found in milk.
Okay, so negative control first.
It's controlled by the lac repressor. Normally, with no lactose around, the lac repressor is active, binds to the lac operator, and keeps the operon switched O-off.
Makes sense. No lactose, don't need the enzymes.
Right. But when lactose is present, some of it gets converted into a related molecule called allolactose. Allolactose then binds to the lac repressor.
Like tryptophan bound the TREP repressor.
Similar idea, but with the opposite effect. Allolactose binding causes the lac repressor to change shape and fall off the DNA operator.
Ah, so the block is removed, the operon can be turned on. That's induction.
Exactly. The presence of lactose induces the operon. But there's a catch. That's where the positive control comes in.
What's the catch?
Bacteria, like E. coli, much prefer to use glucose as their energy source if it's available. It's easier to metabolize. So even if lactose is present, the cell doesn't want to turn on the lac operon full blast if there's plenty of glucose around.
Ah, energy efficiency again. So how does it check for glucose?
Indirectly. Glucose levels regulate the level of a small signaling molecule called cyclic AMP or CANMP. When glucose is low, CMP levels go high.
Low glucose, high CAMP.
This high CAMP then binds to an activator protein called CK, catabolite activator protein. Only when CAMP is bound to it does CAP become active.
So CAP is activated by low glucose via high CAMP. What does active CAP do?
Active CAP binds to a specific site near the lac promoter, and when it's bound there, it helps RNA polymerase bind much more efficiently and start transcription robustly. It's a positive regulator. It cranks up the expression level.
Okay, let me see if I've got this. To get the lac operon fully on in, you need TDO conditions met.
That's the key. Condition 1, lactose must be present to remove the lac repressor. Condition 2, glucose must be absent. So CAP is high, activating CAP to boost transcription.
So it's like ND logic. Lactose, ND, not glucose equals on in.
Precisely. If glucose is present, CAP isn't active, and transcription is very low, even if lactose removed the repressor. If lactose is absent, the repressor is bound and it's off, regardless of glucose levels. It only really fires up when lactose is the best available option.
That's really sophisticated logic for bacteria. And mostly this happens near the promoter, but you mentioned occasional long-distance control?
Yeah, it's rarer in bacteria than in eukaryotes, but it happens. The example given is the NTRC protein. It binds to a DNA site, an enhancer, that can be quite far away from the promoter it controls.
So how does it reach?
The DNA actually loops around, bringing the distant enhancer-bound NTRC close to the promoter-bound RNA polymerase. NTRC then uses energy from ATP hydrolysis to help activate transcription. It shows that looping DNA to connect distant regulators isn't exclusive to eukaryotes.
Fascinating. Okay, so that's bacteria operons, repressors, activators, mostly local control, but some looping. Now let's switch gears entirely. Eukaryotes, like us. Our source says gene control here is, well, way more complicated.
Oh yeah, it's a whole different ballgame. Several big differences. First, as we said, no operons generally. Each gene is usually regulated as its own unit.
One gene, one set of controls.
Right. Second, there are way more regulatory proteins involved, hundreds or even thousands in a typical mammalian cell. And many genes are controlled by combinations of these regulators.
Combinations.
Third, regulatory DNA sequences, enhancers that activate, silencers that repress can be located very far away from the gene's promoter. We're talking tens or even hundreds of thousands of base pairs away, upstream, downstream, even within introns.
Wow, that's really distant. How on earth do they connect? You mentioned looping in bacteria.
Exactly. DNA looping is crucial in eukaryotes too, but it's often mediated by a huge protein complex called, appropriately, the mediator.
The mediator complex. Okay, what does it do?
It acts like a central bridge or scaffold. Activator proteins bound at distant enhancers interact with the mediator, and the mediator, in turn, interacts with RNA polymerase II and the general transcription factors assembled at the promoter. It physically links the remote controls to the core machinery.
So it integrates signals from potentially many different activators bound far away.
Precisely. And repressors can interact with it too to inhibit transcription. It's a key integrator.
Our source also highlighted that eukaryotic activator proteins are often modular. What's that mean?
It means they often have physically distinct parts or domains that do different jobs. Typically, there's a DNA binding domain that recognizes the specific enhancer sequence, and then there's a separate activation domain that interacts with other proteins, like the mediator or co-activators, to actually stimulate transcription.
So one part grabs the DNA, another part calls in the machinery.
Exactly. And you can often swap these domains experimentally. You could take the DNA binding domain from protein A and fuse it to the activation domain from protein B. The resulting hybrid protein would now bind to protein A's target DNA sequence, but activate transcription using protein B's mechanism. It shows they function somewhat independently.
That's pretty cool. Now, a huge difference in eukaryotes is chromatin, right? Our DNA isn't naked like in bacteria. It's wrapped up around histone proteins. That must affect access.
Absolutely. That's a central challenge and a major layer of regulation in eukaryotes. The basic unit, the nucleosome DNA wrapped around 8 histone proteins, can physically block access to promoters and regulatory sequences. DNA is packaged into progressively more compact structures.
So the default state for many eukaryotic genes might actually be off or inaccessible simply because they're buried in chromatin.
That's often the case. So to turn a gene on, you usually need mechanisms to open up or modify that chromatin structure locally.
Okay, how do cells do that? 2 main ways mentioned, right? Histone modification and chromatin remodeling.
Exactly. Let's start with histone modification. The histone proteins have tails that stick out from the nucleosome core. These tails are like little flags that can be chemically modified.
Modified how?
Various chemical groups can be added or removed, primarily on lysine and arginine amino acids in the tails. Common ones are acetylation, adding an acetyl group, methylation, adding a methyl group, and phosphorylation, adding a phosphate group.
And these modifications do what?
They can act in 2 main ways. First, they can directly affect how tightly the histones interact with DNA or with each other. For example, acetylation usually neutralizes the positive charge on lysines, which tends to loosen the chromatin structure, making the DNA more accessible. That's why acetylation is often associated with active genes.
So acetylation helps open things up. What does deacetylation do?
The reverse. Enzymes called histone deacetylases, or HDACs, remove acetyl groups, which allows the chromatin to become more compact and generally leads to gene silencing. The enzymes that add acetyl groups are histonoacetyltransferases, or HATs.
Okay, HATs activate, usually. HDATs repress, usually. What about methylation? Is it always repressive?
No, methylation is more complex. Depending on which lysine or arginine is methylated and how many methyl groups are added, mono, dis, or trimethylation, it can signal either activation or repression. It's very context dependent. For example, methylation on histone H3 lysine 4, H3K4ME3, is usually found at active promoters, while methylation on histone H3 lysine 9, H3K9INE3, or lysine 27, H3P27E3, is strongly associated with silenced regions.
Wow, okay, so it's not just what modification, but where it is and how much. This sounds like a code.
That's exactly the term used, the histone code. The idea is that specific combinations of histone modifications act like signals that are read by other proteins.
Read.
Certain proteins have specialized domains that specifically recognize and bind to particular modified histone tails. For instance, proteins with a bromodomain often bind to acetylated lysines, while proteins with a chromodomain often bind to methylated lysines.
So these reader proteins see the histone modification pattern and then do something.
Exactly. They might recruit other factors that further modify the chromatin or factors involved in transcription, activation, or repression. It sets off a cascade of events. Our source shows a nice example where one modification recruits a reader, which recruits a writer, an enzyme that adds another modification, which is read by another protein, ultimately leading to transcription initiation.
A chain reaction triggered by the initial histone marks. Okay, so that's modification. What about the other mechanism, chromatin remodeling?
Right. Chromatin remodeling complexes are large protein machines that use the energy from ATP hydrolysis. They burn ATP to physically push nucleosomes around.
Push them, like slide them along the DNA.
Yes, they can slide nucleosomes to uncover a promoter sequence. They can also sometimes partially unwrap the DNA from the histone core, or even completely eject histones from a region, or exchange standard histones for variant versions that have different properties. They work with histone chaperones to manage the histones.
So they're like the chromatin architects,
Yeah.
physically restructuring the landscape to grant or deny access.
That's a great way to put it. They make the DNA accessible to the transcription machinery and regulatory proteins, or they can compact it to shut genes down.
Okay, so putting it all together. When a eukaryotic gene needs to be activated, what's the typical sequence?
Well, it often starts with a specific gene activator protein binding to its enhancer sequence, maybe quite far away. This might require some initial chromatin accessibility.
Okay, the activator binds. Then what?
That activator then often recruits co-activators. These can include histone acetyltransferases, HATs, to start acetylating nearby histones, loosening the chromatin.
Adding those open-up signals.
Right. It might also recruit chromatin remodeling complexes to physically slide or reposition nucleosomes, clearing the promoter region.
Making space for the machinery.
Exactly. These changes make the promoter accessible. Then, the activator, often via the mediator complex, helps recruit the general transcription factors and RNA polymerase II itself to the promoter, allowing transcription to begin. It's a coordinated sequence of events.
It sounds like a lot of proteins working together. Our source mentioned transcriptional synergy. What's that?
Synergy is really interesting. It means that when multiple different activator proteins bind to the regulatory regions of a gene, the combined effect on transcription rate is often much greater than just adding up the effects of each activator working alone.
So 1 + 1 doesn't equal 2, it equals like 10 or 100.
Something like that. It allows for very sharp responses. A gene might be basically off with only 1 or 2 activators present, but then switch on very strongly when a third or fourth one arrives. It allows for more sophisticated decision-making based on combinations of signals.
How did that synergy work?
It's thought to involve multiple mechanisms, cooperative binding of factors, multiple simultaneous interactions with the mediator and transcription machinery, maybe multiple chromatin modifying events happening at once. It really ramps up the recruitment and efficiency of the whole process.
Okay, we focused a lot on activation. How do eukaryotes turn genes off using repressors? Is it just the reverse?
There are several ways eukaryotic repressors work. Some are simple. They bind to a DNA site that overlaps with an activator's binding site, just physically blocking the activator competitive binding.
Okay, simple competition.
Others might bind directly to the activation domain of an activator protein, masking it so it can't interact with the mediator or other targets.
Blocking the activator's on signal.
Exactly. Some repressors can interact directly with the general transcription factors at the promoter, inhibiting their assembly or function.
Interfering with the core machinery.
And importantly, many repressors work by recruiting chromatin-modifying enzymes that compact the chromatin. They might recruit histone deacetylases, HDACs, to remove activating acetyl marks.
Closing things down again.
Or recruit histone methyltransferases, HMTs, that add repressive methylation marks like H3K9E3 or H3K27E3. These repressive marks can then recruit other silencing proteins, leading to a more stable, silenced chromatin state. They can also recruit chromatin remodeling complexes that specifically compact chromatin. So multiple ways to put the brakes on.
It seems like regulation often involves these big groups of proteins working together. What are enhanceosomes?
Enhanceosomes are a specific example of that. They are highly structured complexes formed when multiple activator proteins bind cooperatively to closely spaced sites within an enhancer region. Often, architectural DNA bending proteins are involved too. This cooperative assembly ensures that all the necessary components are present together, leading to very robust and precise gene activation, often seen in developmental processes.
A highly organized activation hub. Now, with enhancers potentially working over long distances, how does this cell make sure an enhancer for gene A doesn't accidentally turn on gene B next door?
That's where insulator elements come in, also called boundary elements.
Insulators, like electrical insulators.
Kind of the same idea. They are specific DNA sequences that function as barriers. They can block an enhancer from activating a promoter if the insulator lies between them. They can also stop the spread of repressive chromatin instructions.
So they define regulatory neighborhoods.
Yeah.
Keep the signals contained.
Exactly. They help partition the genome into independent domains so that gene regulation doesn't get scrambled. Proteins like CTCF are often involved in binding to these insulator sequences.
Okay. Now, since eukaryotes don't have operons, how do they coordinate the expression of multiple genes that need to work together, like in a metabolic pathway?
They use a different strategy. Often, all the genes that need to be co-regulated will have the same short regulatory DNA sequence, a response element, in their control regions.
So they all share the same binding site for a specific regulator.
Precisely. Then, a single type of gene regulatory protein, when activated by a specific signal, like a hormone, can bind to this response element near all of those different genes, even if they're on different chromosomes, and turn them all on or off together.
One signal, one activator, hits multiple genes because they all have the right docking site.
The glucocorticoid receptor example makes sense now.
Exactly. It allows for coordinated responses across the genome to a single stimulus.
And this idea of using combinations of regulators, is that the key to creating different cell types during development?
Absolutely. That's combinatorial control, and it's fundamental. You don't necessarily need a unique master switch for every single gene or cell type. Instead, by using different combinations of a relatively limited set of regulatory proteins, the cell can generate a vast number of distinct gene expression patterns, leading to the specialization of different cells.
So cell type A might have regulators 1, 2, and 3 active, while cell type B has regulators 1, 3, and 5. And that unique combo defines its identity.
That's the core principle. It's incredibly powerful and economical. And sometimes there are master regulatory proteins that sit at the top of a hierarchy.
Master regulators. Yeah, a single transcription factor that, when turned on, can trigger a whole cascade of downstream gene expression changes, leading to the formation of an entire cell lineage or even a complex structure.
Like the ILS protein in fruit flies.
That's the classic dramatic example. Expressing the ILS gene topically, like in a fly's leg precursor tissue, can actually trigger the development of a whole eye structure on the leg. It shows how powerful a single master regulator can be in orchestrating a complex developmental program.
Mind-blowing. Okay, this intricate control of transcription is clearly huge in eukaryotes. But there's another layer our source mentions. Epigenetics. What's that about?
Epigenetics refers to changes in gene function that are heritable, meaning they can be passed down through cell division and sometimes even to the next generation, but that do not involve changes in the underlying DNA sequence itself.
Heritable, but not changing the As, T, Cs, and Gs. How?
It often involves those chromatin modifications we talked about, particularly DNA methylation and certain histone modifications that can be copied or maintained when cells divide.
Okay. So patterns of gene activity can be inherited. What's a key example?
A really famous one in mammals is X chromosome inactivation.
Right. Females have two X chromosomes, males have one X and one Y.
Exactly. To balance the dosage of genes on the X chromosome early in the development of female embryos, one of the two X chromosomes in each cell gets randomly chosen and almost completely shut down. Silenced.
Completely silenced, like turned off.
Pretty much. It condenses into a compact structure called a Barr body, and most of the genes on it are not expressed.
And the silencing is permanent for that cell lineage.
Yes. Once that choice is made in an early cell, all of its descendants will inherit the same inactive X chromosome. The active X stays active, the inactive X stays inactive through all subsequent cell divisions.
How does it remember which one to keep off?
It involves a long non-coding RNA called ZIST, which is expressed only from the inactive X. This RNA coats the chromosome and recruits protein complexes that modify the histones and methylate the DNA, establishing a stable, heritable, silent state. It's a classic epigenetic mechanism.
Wow. Okay, what about DNA methylation itself, more generally?
DNA methylation, the addition of a methyl group, NACRECH3, to cytosine bases, specifically in CpG sequences in mammals, is another major epigenetic mark. High levels of methylation in a gene's promoter region are very often associated with long-term gene silencing.
How does methylation silence genes?
It can work in a couple of ways. Sometimes the methyl groups directly interfere with the binding of activator proteins. More often, methylated DNA recruits specific proteins, methyl CpG binding proteins, which in turn recruit other factors like HDACs and HMTs that create a repressive chromatin environment.
So methylation flies the DNA for silencing machinery, and this can be inherited.
Yes, that's key. After DNA replication, the old strand still has its methylation pattern. Enzymes called maintenance methyltransferases recognize this pattern on the hemimethylated DNA and copy it onto the newly synthesized strand. This ensures the methylation pattern and thus the silencing is passed down to daughter cells.
So the memory is maintained. And then there's genomic imprinting. That sounds related.
It is. Genomic imprinting is a fascinating phenomenon where a small number of genes are expressed only from the copy inherited from one parent, either mother or father, while the copy from the other parent is epigenetically silenced, usually by methylation.
Wait, so for imprinted genes, it matters whether you got the gene from mom or dad.
Exactly. The silencing imprint is established in the sperm or egg cells and then maintained throughout development. The UGF2 gene is a common example typically, only the paternal copy is active, the maternal copy is imprinted, silenced. It often involves methylation affecting insulator elements.
That adds another layer of complexity to inheritance. And these epigenetic patterns, can they be influenced by like our lifestyle or environment?
That's a really hot area of research right now. There's growing evidence that environmental factors, diet, stress, toxins can influence epigenetic marks like DNA methylation over time. Even in identical twins who start with the same DNA, their epigenetic patterns can diverge as they age, potentially contributing to differences in health and disease risk.
So it's not just the DNA sequence, but how it's marked and packaged, and that can be dynamic.
Okay, we've covered a ton on transcriptional control and epigenetics. But regulation doesn't stop once the RNA is made, right? Post-transcriptional control.
Absolutely not. There are still many opportunities to control gene expression after the initial RNA transcript is synthesized.
Like what?
Well, think about RNA processing in eukaryotes. How the RNA is capped at the 5-foot end, how the poly-A tail is added at the 3-foot end, and especially how it's spliced. Then there's RNA editing, where the sequence of the RNA itself can be changed.
Splicing. Cutting out introns, joining exons.
Right. Then controlling which mRNAs get exported from the nucleus to the cytoplasm. Once in the cytoplasm, where does the mRNA go? Some are localized to specific regions. Then controlling if and how efficiently the mRNA is translated into protein by ribosomes. And finally, controlling how long the mRNA lasts before it's degraded. Each step is a potential control point.
So many dials to tune. The source really emphasized alternative splicing, though. Yes. Alternative splicing is a huge deal, especially in complex organisms. It means that a single gene, with its multiple exons and introns, can be spliced in different ways to produce multiple different mRNA molecules, and therefore multiple different protein variants or isoforms from that one gene.
So from one gene blueprint, you can build several different versions of the protein product.
Exactly. You can skip exons, include introns that are usually removed, use different splice sites. It massively increases the coding potential of the genome. You get protein diversity without needing vastly more genes.
Is this common?
extremely common in mammals. Most human genes are thought to undergo alternative splicing. The source gives that incredible example from the fruit fly, the DSCAM gene.
DSCAM. What's so special about it?
Through complex alternative splicing of different potential exons, that single DSCAM gene can generate something like over 38,000 different protein isoforms.
38,000.
Yeah.
From one gene.
Yep. And this diversity is thought to be crucial for helping neurons wire up correctly in the fly's brain, giving each neuron a unique identity tag. It's a stunning example of how post-transcriptional control can generate immense complexity.
That really puts an exclamation point on the idea that gene control is multi-layered. Okay, let's just try to wrap this up. We've gone deep today.
We really have. The big picture is that controlling which genes are expressed, when, where, and how much is absolutely fundamental to life as we know it, especially for complex organisms.
It's how cells with the exact same DNA library can become so incredibly different: muscle, nerve, skin, whatever.
Right, and this control happens at so many levels. Transcription initiation is a huge one, especially in eukaryotes with all the complexity of chromatin, histone modifications, remodeling complexes, enhancers, mediator.
And the gene regulatory proteins, the transcription factors, binding to specific DNA sequences using motifs like HTH or zinc fingers.
Then there's epigenetics, DNA methylation, exon activation, imprinting, allowing patterns of expression to be inherited without changing the DNA sequence itself.
And even after the RNA is made, post-transcriptional controls like alternative splicing add yet another layer, massively expanding protein diversity from a limited number of genes.
It's an incredibly intricate and elegant system.
And understanding it isn't just, academic. It's crucial for understanding development, basic cell biology.
And also what goes wrong in diseases. So much of cancer research, for example, revolves around understanding how gene expression control gets messed up.
Absolutely. So maybe something for everyone listening to think about. That journey from a single fertilized egg with one set of DNA to the trillions of specialized cells in your body right now, it all hinges on this incredibly dynamic, multi-layered control of gene expression we've been talking about.
And maybe consider how fragile it can be, too, how disruptions in these pathways, whether genetic or maybe even environmental influencing epigenetics, can lead to disease. It really makes you appreciate the precision involved.
Definitely.
Yeah.
Perhaps something specific caught your ear today. Maybe the histone code or master regulators or how bacteria use operons. It's a vast field, always more to explore.